Good afternoon. Thank you very much for inviting me to this event. In my group, we're extremely interested in using different kinds of spectroscopy and microscopy to learn about the diffusion and viscosity inside the cells. However, a few days ago, I looked at the program and I realized that actually I'm speaking in the DNA session. So I had to slightly change the title of my talk. And so I had to remove the temperature, and instead now I'm going to talk about using our probes called molecular rotors for probing microscopic viscosity in live tissues and cells, but also probing nucleic acid conformation in a dynamic way. I'll still stick with molecular rotors first because it describes the background and describes the main principles of how our approach and our strategy works. So our interest comes from the fact that cells and tissues are extremely heterogeneous environments. There are lots of different organelles. Most of them are surrounded by hydrophobic membranes, and all of this is embedded in essentially aqueous phase of the cell or cytoplasm. So in this really complex medium, the distribution of these organelles and the hydrophobic properties of these organelles, so in other words, their viscosity, play a large role in controlling lots of cellular processes. And there's evidence for links between viscosity and disease and malfunction, and also processes such as bimolecular reactions and transport will, of course, be affected by viscosity. However, measuring viscosity on a single cell and organelle level is a really challenging process. And there are lots of techniques that have been developed over the years, such as FRAP and FCS and anisotropy methods. However, when we started this work, we realized that most of these methods have two major limitations. And one is that most of them are single point measurements. So they probe one single point at any one time, and that doesn't give you an image or the distribution of how viscosity can be changing in different parts of the cells. And the second problem we noticed is that some of these techniques require continuous accumulation of data. So if this accumulation takes time, then you'll end up with averaged values of viscosities that you measure. And so ideally, we want a method that measures dynamic changes. So what I would like to try to persuade you today that our method, based on fluorescence of so-called molecular rotors, overcomes both of these problems. And so that can provide us images with good spatial resolution, and these measurements are fairly fast. So we can actually monitor dynamic processes within the cells as viscosity changes within them. Okay, so what are molecular rotors? They're small synthetic fluorophores in which there's a large geometrical conformational change that actually leads to a large change in their electronic structure. So they go from the bright emissive state to a dark non-emissive state via the rotation. And of course, the speed of their conformational change or the rotation is strongly affected by the viscosity of their surrounding medium. So, for example, in a non-viscous environment, this molecule, which is a Bodepie molecular rotor, has the phenyl ring rotating around the core of the fluorophore. The rotation of this unit leads to deactivation of the bright excited state of the Bodepie into a dark state. And as a result, the fluorescence intensity is really low, and also fluorescence lifetime is really short as well. So when you now increase the viscosity of the environment, rotation is restricted, and as a result, fluorescence intensity and the fluorescence lifetime goes up. So in our group, we really prefer to use methods based on fluorescence lifetime of our probes, because intensity, as you can imagine, depends on how much fluorophore you have in a particular part in the cell. And in a complex, heterogeneous environment of cells or biological molecules, you simply can't predict the uptake of your probe in any particular place of the organelle. And so this fluorescence intensity measurement is biased by how much of your probe is there. However, the lifetime is an intrinsically ratiometric method. And so it measures the decay of our excited state, that is concentration independent. And so hopefully as a result, we'll be able to use so-called fluorescence lifetime imaging microscopy, or FLIM for short, to quantitatively measure viscosity in a cell. So plotted here is the lifetime, it's the measure how fast the fluorescent state decays, versus viscosity. And for one of our favorite membrane-based rotors, Bodepie, you can see that there's a really good correlation with large dynamic range over a couple of orders of magnitude of viscosity. And we believe that this is a biologically relevant range, which will be of interest to our measurements in cells. Okay, so we can quantitatively measure viscosity using the technique called FLIM, where we measure fluorescence lifetime decay in every single point of our image. And then we fit those decays and plot the lifetimes. And in case of molecular rotors, lifetime means viscosity. Okay, so what do we do in my group? Well, we design and synthesize new rotors. We collaborate with others who provide rotors to us. And then we use them for a large range of various problems where diffusion or crowding or conformation might be important, but can't be measured easily using other spectroscopic methods available. So of large importance to us is cellular membranes, things like lipid rafts and diffusivity that is associated with disease. Organelle viscosity, measuring viscosity in vivo, again correlated to disease. And obviously our focus to my talk today is also things like conformation of nucleic acids and aggregation of proteins. Okay, so let's start again by one of my favorite rotors, Bowdesby-based, and how we measure viscosity with it in lipid membranes of various origin. So we have our rotors that, due to its hydrophobicity and its structure, incorporates seamlessly into monolayers or bilayers of biological membranes. And then we can perform confocal or multiphoton imaging of the rotor incorporated in the membrane. And of course, FLIM, where the decay, let me just repeat, in every single point of this image is measured. And so as a result, the lifetime here, so blue for shorter lifetimes and red for longer lifetimes, essentially visualizes different domains where the lipids are packed in a particular way. And that gives us the map of viscosity with good resolution. Okay, so when we add our rotors to cells, if we design them in the right way, they internalize and they can stain certain organelles. In this case, endosomes are stained in this HeLa cell line, and we can record the fluorescence decay, which then can be calibrated to a particular viscosity value. So here the value is about 140 centipoise, which is 100 times higher than that in water. So this was a good discovery for us now many years ago, but essentially we know that we are probing lipid-based domains where viscosities are 100 times higher than that of water, and diffusion control processes are really slowed down compared to what will happen in water. Consequently, we design rotors that have positive charge at the end of their tail, and that acts as a stopper, and that prevents endocytosis of our dye inside the cells. And that means that we can actually stain plasma membrane of our cells, which is really useful because plasma membrane packing and viscosity is responsible for a plethora of different cellular processes. And as it turns out, it's important in things like stem cell development. It's important in packing of cells in 3D culture, such as spheroids. And it's also important during chemotherapy. So, for example, shown here is the data of how this, plasma membrane viscosity changes in response to cisplatin treatment. And we can also perform mass spec analysis of the lipids extracted from these treated cells at every point of treatment, and back up our viscosity data with lipidomics analysis from the same culture, as well as study chemoresistant cells that are grown separately and studied separately. Also, sort of a side effect of these two positive charges is that now our rotor is really water-soluble quite well, and that means that we can use it in animals without high unwanted uptake and things like liver and kidneys. So it has extra degree of biocompatibility. So far, I talked about our rotors sort of passively diffusing into the cell and then going into certain organelles because of their hydrophobicity or lipophilicity properties. But if we want a particular organelle targeting, we can actually do better than this. And so this is an example of halo-targeted rotor, where our rotor has been conjugated to this haloalkyne ligand, which can interact with targeted haloprotein, which is expressed specifically in a particular organelle. And so here shown are maps basically obtained using the same kind of rotor dye, but targeted, or untargeted here, or targeted to either ER or mitochondrial matrix. And it turns out this is quite useful for looking at certain diseases. So earlier in the talk, we heard about alpha-antitrypsin. And so we use this particular ER rotor to study abnormalities in alpha-trypsin when it goes from normal A1 form to Z-antitrypsin, which it turns out is accompanied with large increase in crowding and viscosity. Okay, one thing I mentioned straight at the beginning is that we want to measure dynamically how viscosity changes in cells. And turns out this is quite useful, it's quite easy to do using this approach, because each of these images doesn't take that long to accumulate. So by consecutively recording film images, we can now study dynamic changes in viscosity during some useful processes. So I already mentioned chemotherapy. And here's another example, when we actually subject our cells to production of CO. So this is an unusual rotor because now it's attached to a ruthenium center. And the special property of this transition metal complex is that this first complex, even though it has the rotor attached to it, is completely dark. However, in the presence of carbon monoxide, it reacts really fast and irreversibly with carbon monoxide quantitatively. And as a result, the electronic structure of this complex changes a lot, and it becomes fluorescent. But what we realized is that the molecular rotor attached to the complex retains its molecular rotor structure, function, and the lifetime of the rotor does not actually depend on whether the complex is bright or dark. So we now have two parameters. One is fluorescence. So bright signal means that we have CO present, and lifetime is indicative of the viscosity of the environment. So what we did here is that we produced CO in cells in two different ways. One is using a chemical comb, which just releases CO in contact with water. And another one is by incubating cells with heme, which actually triggers biochemical pathways that cause inflammation and CO release this way. And so in both of these cases, I hope you can see that fluorescence goes up, so CO is being produced, but lifetime, and so viscosity behaves in a very different way. So the lifetime produced by comb is stable. So CO is being produced, but nothing happens in the cell. Viscosity is not changing. But in the presence of heme, which we take as a model for inflammation in some crude kind of way, we see a reduction in lifetime, so a reduced viscosity, which we now think is associated with radical production of these conditions. Okay, so now onto DNA. So, so far, all the rotors that we use were freely rotating in the medium. And this is how we use their function to measure the viscosity of their surrounding environment. So this is the prerequisite, that they never bind the molecules that we want to study. However, with DNA, we now want to use the property of the molecular rotors to bind a particular DNA conformation. And it's the different structure upon binding to DNA which will tell us about this presence of different structures. Now, most of you will know that DNA is present in the cell in the most abundant form of the double helix of bDNA, which is then tightly packed in chromosomes. But occasionally, this double helix needs to unwind, and then the single strands of DNA and RNA can undergo different dynamic conformations, such as triplexes or quadruplexes. And so, of particular interest to us is this unusual and sort of not very abundant conformation of DNA, which is called the G-quadruplex, which happens when single-stranded DNA is rich in guanines, and then guanines can participate in Hooke's-Steen hydrogen bonding with each other, forming this tetrad. And so we are going to use the ability of our molecular rotors to specifically bind on top of this tetrad, and by the specific conformational change, cause a change in lifetime, and then indicate their presence in cells. Now, why do we want to measure G-quadruplexes? Obviously, duplex is a lot more abundant. However, as I said, during lots of different processes, such as replication and transcription and in telomeres, the double helix does not form, and instead, guanine-rich regions of DNA are present, and there are lots of putative sequences that are suspected to form this unusual G-quadruplexes. And in fact, there's a lot of data that they can regulate cellular processes at these really essential points of cellular processes. So by detecting the quadruplexes' presence and their role in these processes, we can get really important sort of quantitative biology information on their presence. However, it's quite tricky to detect them because bDNA, the duplex DNA, is very, very abundant. So really the probes for quadruplexes are really lacking. So this molecule here is a so-called triangulenium because of its triangular structure that has two arms with those secondary amines called morpholino groups. that can bend in and out of the plane of the triangulenium, and that gives us the molecular rotor properties. So if you place this molecule in the viscosity in the environment of different viscosity, its lifetime will change drastically from low to high viscosity. However, specifically for this project, what's of interest is that this molecule intercalates into the double helix DNA, and that allows the arms to be quite close to the proximity to the core and quench the emission of it. However, if it binds to quadruplexes, the arms are participating in hydrogen bonding with loops and grooves of quadruplex, and are taken away from the core, and that produces a really long fluorescence lifetime and really high emission intensity. So now we have very different lifetime signature upon binding to duplex and quadruplex DNA. And we confirm this by studying the large panel of different single-stranded duplex or quadruplex topologies. And so now we can record FLIM, fluorescence lifetime images, of our dye called delta M2 in cells. And here the low lifetimes indicate the complex bound to a duplex, and long lifetimes indicating the binding to the quadruplex. Now, this is a nice concept, but what can we do with it? So imagine we incubated our cells with our delta M2. We see a certain lifetime. This lifetime, typically in the cell, will be between duplex and quadruplex, because it's bound to everything, and our resolution, which is diffraction limited, is not enough to distinguish individual quadruplexes. So you might say, which uses this? Well, what if we affect our cells in some way? So for example, if we add a ligand to be screened, remember quadruplexes could be some potentially interesting therapeutic targets, including for cancer. So if the ligand is strong binder, and if its concentration is high enough, it will displace our delta M2, and we'll see a change, a reduction in lifetime. And that's exactly what we see here. So cells incubated with delta M2, and when we add the pyridostatin, which is a known strong quadruplex binder, we see a large reduction in lifetime seen from cells, and no change when we use DAPI, which is a known intercalator of the DNA. So this is really nice because actually we can use it as an additional verification that our dye was bound to G4 in the first place, because when we added G4 binder, our dye has been displaced effectively, but also it's a nice screening platform, because we can use non-fluorescent ligands that might bind to quadruplex, might penetrate into cells or not, but our displacement assay, FLIDA, can tell us whether they displace our dye from the quadruplexes in the cell or not. Okay, so this is another biological example where now we use the lifetime of our delta M2 in cells to test whether certain enzymes, helicases, affect the quadruplex production within the cells. So there's a lot of evidence that helicases such as FANJ or RTEL effectively unwind quadruplexes at telomeres. But this is only secondary evidence from secondary biophysical studies. There's no direct evidence in cells. So what we can do is we can take our cells incubated with delta M2 and either produce the mutants that don't have FANJ or knock down FANJ from different types of cells. So the consequence of this is no helicases are now existing in the cells. And so quadruplexes are not going to be unwound. There'll be more quadruplexes, and so the longer lifetime is observed. And that's exactly what we see in our delta M2 experiments. So our experiments, again, confirm the function of helicases such as FANJ or RTEL in cells. And we can do FANJ mutants and knockdowns, RTEL mutants and knockdown, and then even produce a mutant that doesn't have either of these two helicases, and then have an additive effect of increasing lifetime. Okay, so have time. So this dye that I talked about was a fluorescent molecule. So this is great, but if you want to use your probe alongside, let's say, GFP or another common fluorescent marker, then you might want to do some multiplexing to allow you to image, let's say, FRET using fluorescent marker, and quadruplexes using some other technology. So we decided to try to look at the phosphorescence, which is a similar phenomenon to fluorescence, except it involves triplet state decay. And as a consequence, it's about 100 to 1,000 times longer. So it happens at a completely different time scale. So transition metal complexes are really, really good candidates for phosphorescence because they have efficient transfer of the singlet to triplet states. And so we synthesized these platinum complexes, which were known to be duplex and quadruplex binders. All of the complexes that we've made have a large increase in their fluorescence upon binding to G4 and duplex DNA. But luckily, this one series of complexes with chloride ligand attached to the platinum also have a good distinction in lifetime. So quadruplexes have a much longer phosphorescence lifetime compared to a duplex, and both of them are much longer than the V complex in solution. So shown here is the screen for all the complexes, just to show that here's the duplex lifetime, about 700 nanoseconds, and quadruplex lifetime, about 1.6 microseconds. So 1.6 microseconds is more than 100 times longer than what we've seen for delta. So if you want to do multiplex measurements, you can use your fluorophore or autofluorescence in fluorescence channel and have a completely different channel for phosphorescence. How does it work in cells? So we can see phosphorescence from our dye from various different places in cells, but it's most intense in the nucleus. And we can also see the lifetime from cytosol, nucleus, and specifically nucleoli. And all of them are in the region where we know that their quadruplex is in there, because duplex DNA comes at about 800 nanoseconds. So anything which is longer will be to do with quadruplexes. And now again, remember our FLIDA assay, so displacement assay, just to confirm to ourselves as well as to others that we really do see quadruplexes. We now add a really strong G4 ligand, nickel sulfane, which is supposed to kick all of our complex from quadruplexes. And fair enough, we see a reduction in lifetime upon addition of nickel sulfane, so characterized by green color here. So our dye was bound to quadruplexes, but it's not anymore. Okay, do I have time? Or? 2 minutes, okay. So let me just very quickly describe to you our efforts to try to make better dyes. Yeah? So this technique works really, really well, but the trianguleniums, which perhaps to date is the most useful dyes for G4 imaging, have lots of good qualities, but they're not very bright. They have low quantum yield, so you actually need a long time to accumulate the images. So we want to make better dyes. The dye development process is very laborious. You have to make the molecule, you have to purify it, have to test its physical properties, test if it goes to cells, and then start again if it didn't work. So one of my really talented students, Atika Majid, came up with a platform for high throughput synthesis of potentially better probes on the basis of our triangulenium 4. So we now have triangulenium with azide attached to it, which will react readily with alkynes. And this is a copper-catalyzed click reaction, which is very efficient, no purification required, and is biocompatible. So Atika purchased all of these amines with alkynes attached to them, and then performed the automated synthesis. I hope you can see here, the robot is doing the synthesis in 96 well plates. And then a different robot can do mass spec and NMR of all these reaction mixtures. And if the reaction didn't work, we simply discard this compound and do not promote it to the next step of our high throughput synthesis and investigation. What Atika found, really unexpectedly, is our probes are now more emissive, but also they have this really bizarre property that quadruplex enhances its emission in lifetime, but duplex actually quenches it. So that's really unusual, but it's really beneficial, right? If we quench duplex, we'll have a better signal from quadruplex. So our next level of screening is for compounds of this series to have this unusual switch-on, switch-off behavior. And if we don't see it, we don't promote it to the next stage. Okay, so now it's going to be really quick. I'll just show you a beautiful illustration from Atika, how she started with all these amines, discarded the ones that didn't work synthetically, then discarded the ones that didn't go through the lifetime screening, and then discarded the ones that didn't go to cells, and finally she ended up with just one compound, which we can take into cells, do FLIM, and now we're in the process of verifying if it's any good for any biological studies. Okay, so this is me. I hope I persuaded you that molecular rotors are really useful probes for viscosity, crowding, and even nucleic acid conformation. We can monitor dynamic processes. We can do quantitative measurements inside cells and tissues and other biological objects because we do FLIM. And we can image temperature as well, but I didn't have time to talk about this. And all the credit goes to all these wonderful people in the group who did all this work over many years. Thank you for your attention.
questions for Marina? Okay, maybe I'll start then. So it was interesting you were talking about measuring viscosity in different cellular compartments. Do we understand what the roles of different values of viscosity in different cellular compartments might be for normal cellular function?
That's a really good question. So we understand how viscosity values correlate to diffusion in membranes, because early on, when we first developed this method, we need to do lots of control measurements to persuade others that we're measuring the right values. So basically, our viscosities translate into diffusion coefficients that people would expect from a particular viscosity value for a particular size solutes. So they have really mean sort of value to them. When we look at crowding systems, so aggregating proteins, or let's say crowding response in the ER of the cell, this is more of the gray area, because there we are probing solvent-free volumes when protein residues come together and restrict the rotation of the rotor. So there we'll be a lot less confident translating our viscosity using Stokes-Einstein diffusion coefficient. I don't think it will work. But what we make sure does work is high viscosity means lower diffusion at all times.
For the organelle halo tag concept you showed, is the dye still integrated into the membrane, or does it stay outside?
It's a really good question. So we modeled our halo tag rotor construct, and you can have halo tag tags of different lengths as well. And so what we made sure is the rotor doesn't stick on top of the protein. So we had cases when the rotor just quenches because it interacts with one or two amino acids just outside the pocket, binding pocket of the halo tag, and then it doesn't measure viscosity, it measures interaction. So our dye sticks into the aqueous environment because the tail is really short, and it measures the viscosity of the aqueous environment of the mitochondria and ER. But if you want to measure membrane, what you then have to do is to make a really long tail, which allows the rotor to go out of the protein and insert back into the membrane. And people like Stefan Mathieu did it with their flippers. We haven't done it yet.
The reason why the dye on its own, when you infuse the cell with it, doesn't go to other membranes is simply because it has a much higher affinity.
You mean the halo one? I think we were just lucky with Bodipy. If you make any rotor based on cyanine dyes, it will just have non-specific binding everywhere because its hydrophilicity is such that it prefers to go to mitochondria no matter where you target it. But Bodipy somehow is driven by this halo tag interaction. I guess maybe it might end up in other parts of the cell, but it probably is not bright enough because viscosity is low. So what we see really beautiful staining corresponding to the organelle of desired targeting.
Hi, thank you very much for a lovely talk. You mentioned at the end very briefly about simultaneous measurement of viscosity and temperature. I would imagine in simple solutions these two things would be very well correlated. I just wondered if you'd seen in a cellular context where those become completely uncoupled. Have you seen any examples of that?
So you're completely right that they're coupled. And they're particularly coupled in a viscous environment, right? Because temperature will affect the viscous environment a lot more than it affects the non-viscous. So water hardly changes in viscosity when you change the temperature. But the membrane changes from, say, 30 centipoise to several hundred centipoise just, between sort of 15 and 37. So we have the dyes for simultaneous measuring of viscosity and temperature, which is quite tricky, actually. It has to have two readouts from the same dye. But unfortunately, this kind of dyes, they also produce things like reactive oxygen species, so we can't use them in cellular context. So in cellular context, we have dyes that just measure viscosity or just measure temperature.
I think we're done with all the questions, so we'll thank Marina again.